A very good morning to Dr. Nasrul. My name is Shalini Sandiran, matrix number 211151348. So today I'm going to be presenting on the introduction of laser-induced fluorescence, which is used to detect cancer. So what is laser-induced fluorescence or LIF? So this LIF is a spectroscopic method that uses laser light or laser beam to excite molecules and measure the emitted fluorescence. This technique is also rapid, non-invasive, and sensitive to detection of cancerous tissue based on their fluorescence characteristics. So we also can use this laser-induced fluorescence to identify and quantify different types of molecules such as proteins, DNA, and many more. So how does this LIF work? So it involves three main steps, which is excitation, emission, and detection. So during the first process, excitation, when a laser beam with a specific wavelength is directed at a sample, causing some of the molecules to absorb photons and jump to a higher energy level. So during the second step, which is emission, the excited molecule, it returns to a lower energy level while releasing the photons with a different wavelength than the laser beam. So during the final step, which is the detection part, so the emitted fluorescence, which is collected by the photodetector, such as photomultiplier tube, and it is analyzed by a spectrometer. So next is why we use LIF to detect cancer. So we use this laser-induced fluorescence, which can distinguish or differentiate the cancer cells from normal cells based on their fluorescence spectra, intensity, and lifetime. LIF can also detect cancer biomarkers such as tumor-specific proteins or DNA mutations by using fluorescent probes. That's all from me. Thank you.
Assalamualaikum and a very good morning. My name is Nur Aizati bin Rafi with a metric number 201151407. So today I will be present about the laser induced fluorescence method, LIF method. So biopsy is known to be universal method disease detection, but due to the complicated situation, it gives inaccurate diagnosis. After a few previous years, the optical techniques called laser-induced fluorescence (LIF) was introduced. So there are three methods. The first method is utilize exogenous or endogenous fluorophores and then detected the appropriate anatomical sites by fiber optic probe in their trials. After the encoding by laser with specific wavelength, they will differentiate the normal and cancerous tissues according to the fluorescence intensity, the wavelength of the fluorescence or the fluorescence intensity ratio of primary and secondary peak for maximum accumulation in tissues. This method belongs to single point diagnosis. The second method is used by many other groups is that they utilize exogenous or endogenous fluorophores and an endoscope-based system bonding with laser-induced fluorescence detection system and an optical imaging system to get the image of the whole tissues. Therefore, such a method can be called imaging diagnosis. For instance, OCT is usually taken advantage as the imaging system to visualize mucosal thickening or abnormal massive development over the imaging time points. And lastly, the third method is time-resolved autofluorescence spectroscopy by laser-induced fluorescence was commonly applied in cancer diagnosis as well. So it based on the measurement of the spectrally resolved transition decay time of laser-induced autofluorescence emission from different tissue. For example, via detect the fluorescent lifetime to differentiate the tissue. So this method is not relevant to the scattering of detection environment and the alternation of optical system, but because they cannot identify the location of the cancer. That's all from me. Thank you.
Hi, my name is Elisha, my son of Mugudan. My metric number is 2111511284. So today I would like to present about the LIF application. So welcome to the world of biomedical fluorescence. Today we would like to explore how the fluorescence transform our understanding to cellular of and molecular process. So the first is about cellular imaging. What is a cellular imaging? Cellular imaging is a divide into the live cell imaging, and fluorescence provides unveiling dynamic cellular structure under the microscope. Next is molecular biology. which is explore the DNA or RNA labeling and fluorescence protein revol- revolarization technique like FISH and PCR. The next is flow cytometry, which is witness cells shortening and analyzed through the fluorescence dyes, a changer in a study specific of cell population. Next is in vivo imaging. Vivo imaging is to discover how the molecular imaging with the fluorescence properties allow us to the violent visualizations biological process in living organism. Next is about the diagnosis. The diagnosis is uncover biosensor and immunofluorescence axes aiding in the disease detection and protection quantification for the diagnosis. Next about the photodynamic therapy. It is about the explore cancer treatment possibility through the photodynamic therapy, leveraging fluorescence and agent affected by the lightning. Next about the neuroscience. Neuroscience is the delve into the fluorescence in neuroscience from cell-kill imaging of the neuron transit unraveling of the mysteries of the brain. And the next one is a drug discovery, which is the wrap up with the impact of the fluorescence in drug discovery, speeding up high the throw-up screening for the potential drug candidates. The last is about the conclusion. So by these things all, in the conclusion, fluorescence in the biomedical is a powerful tool shaping in the future of the healthcare and the science of the discovery. Thank you.
This is a method of laser-induced fluorescence diagnosis for stomach tumor using 5-ALA photosensitizer, a spectral system and a video system to measure the fluorescence signals from the stomach tissues after oral administrations of 5-ALA, which induce a accumulation of PPIX, a natural photosensitizer in the tumor cells. A fluorescence signals were excited by a lasers with a wavelength of 632.8 nanometer and detected by a spectrometer or a camera. Their fluorescent intensities and contrast were used as the identity and differentiation tumorous tissue from a normal tissue. In this, research also found that both systems were effective and provide optimal conditions for the diagnosis, such as the dose and timings of photosensitizers administrations. The methods could also help surgeons to conduct biopsies, laparoscopy, and intraoperative assessments. The method also could help surgeons to conduct biopsies, laparoscopy, and intraoperative assessments of tumor extensors and detect of a cancerous focus. In this method, a novel fluorescence probe for the detections of hydrogen sulfide in living tissues and cells. The probe is based on a coumarin derivative that can react with hydrogen sulfide to produce a strong fluorescence signal. The fluorescence signals can be measured by the fluorescence spectrometers or a fluorescence micrometer or a fluorescence microscope. The probe has a coumarin core with a 4-nitrobenzene group at the 7th positions and the alkyl group at the 3rd position. The 4th nitrobenzene group acts as a contrast for a coumarin fluorescence, while an alkyl group acts as a reaction site for hydrogen sulfide. When probe encounters hydrogen sulfide, the alkyl group undergoes the thionein reactions, resulting in the formations of the Theodorus bond and the release of the 4-nitrobenzene group. This process restores the fluorescence of the coumarin core, which can detect by the fluorescence spectrometer and a fluorescence microscope.
Okay, for my part, which is a conclusion of this report, from which is what can I conclude is laser-induced fluorescence, or as known as LIF, is a molecular emission spectroscopy method used for detection of cancerous tissue. This process involves using visible ultraviolet light that stimulates molecules in the sample by making them absorb photons and fluorescence. There are three advantages that I can conclude, which is LRF help identify cancerous lesions with a very high degree of accuracy. It also helps in live imaging during surgeries and allows surgeons to see cancerous tissues from healthy ones. And lastly, it is a non-invasive nature, which is avoiding the necessity of invasive techniques like biopsies in some situations. However, the accuracy of LIF measurements can be influenced by variation in tissue composition, heterogeneity within tumors, and possible influence of surrounding structures. This challenge requires further research and technological improvement to increase the reliability of LIF in clinical settings. That's all from me. Thank you.